Hi everyone, I'm Sawani Talekar. I'm one of the senior scientists at Crystal Pharmatech, and I'm going to talk about the crystal structure prediction and PBPK modeling assisted experimentation. Compound A was provided to us. 400 polymorph screening experiments were conducted on this. A single slow evaporation experiment of these 400 yielded crystals that were suitable for single crystal X-ray diffraction. And a unique pattern was found. The simulated pattern from the single crystal can be seen here in the blue trace as compared to our starting material, which is form 2, shown in the red trace here. And as you can see, they're very different from each other. So it was interesting that we found one unique form out of these 400 experiments. So additional experiments were set up to reproduce this unique form. However, we were unsuccessful. So the question we had was, if this form is not reproducible, how does this impact on the further development of this compound? And just to note, compound A has needle-shaped morphology, as shown here in the PLM image. So the important decision here was to know the thermodynamic relationship between form 1 and form 2. So we relied on the polymorph prediction suit. Polymorph prediction indicated that form 1 is more stable in comparison to form 2. Comparatively, form 2 we know is much easier to crystallize. So 50 non-conventional crystal growth experiments were performed. These included polymer-induced crystallization. Now, in polymer-induced crystallization, we use fine mixtures of polymers or polymer beads, which act as a surface for nucleation of the crystal. Capillary crystallization was another technique that was utilized, in which fine capillaries are used to slowly evaporate the solvent and allow nucleation of the crystal. Gel crystallization was another technique that was included, and in this, we dissolved the compound in a bunch of solvents where gel was obtained, and the gel was then subjected to different temperature and humidity conditions to probe crystallization of this form 1. On the bottom left, we can see the relative energy versus density graph, which depicts the different polymorphs predicted for this compound. The blue dots you can see are all of the different polymorphs. Form 2 is shown in the purple box, and form 1 is shown in the black box. Now, you would ask, how do I know this is form 2 and the other is form 1? With the AI-based polymorph prediction, we can also get a simulated X-ray pattern, which can then be compared with our experimental results. And that's how we know which is form 1 versus which is form 2. So with these 50 non-conventional crystal growth experiments, capillary crystallization was able to give us form 1 consistently, and we were able to make it using capillary crystallization reproducibly. The PLM image shown here shows that form 1 has a block-shaped morphology as compared to the form 2, which is our starting material, and had needle-shaped morphology. On the X-ray diffraction overlay here, we see the blue trace, which was the very first experiment that got the unique form 1, and the second red trace here, which is basically the X-ray diffraction pattern of our crystals obtained from capillary crystallization. And as we can see, it's a pretty good match. So we know that form 1 is more stable as compared to form 2. But what is the relationship between form 1 and form 2? Is it monotropic, meaning one polymorph will be more stable than the other at every given processing temperature? Or is the relationship enantiotropic, meaning that one polymorph will be more stable than the other at a specific range of temperature, and the other polymorph will be more stable beyond this range? So to find this out, we did competitive slurry experiments between form 1 and form 2 in our processing solvent at temperature starting with 5 degrees Celsius all the way up to 80 degrees Celsius. And at each of these temperatures, we found that form 1 is the most stable form as compared to form 2. So the relationship between form 1 and form 2 was concluded to be monotropic. We know that form 1 is more stable as compared to form 2, so it is going to have lower exposure, but what is the impact on the FIH predictions? So what we did is we did PK modeling for Form 1 and Form 2 at 2 different dose levels, 50 mg and 100 mg. And as you can see from the plasma concentration versus time profile on the right, Form 2 clearly shows superior exposure. And this is because Form 1, being the more stable form, has lower solubility as compared to Form 2. And as we can see from this graph, Form 2 is showing a 2-fold difference in the Cmax as compared to Form 1 at both the dose levels. So the question then was, do we need a formulation-enabling technology, such as the nanosuspension or salt or co-crystal form, or even amorphous solid dispersion, for giving the boost in solubility for Form 1? So the takeaway from this case study is that the crystal structure predictions can help us design experiments more efficiently to make the most stable form reproducibly, and PBPK modeling gives us an idea of the formulation strategy that is necessary to reach the desired exposure in humans.